Hello, we are the group that is working on the melanoma cancer. To start off, we want to know what melanoma is, how is it identified, and how is it treated. So a quick summary of the disease, we want to know what melanoma looks like. Melanoma comes in like a bunch of different shapes and forms and sizes, so it isn't a one-size-fits-all type of thing. Everyone has it differently. Sometimes it appears as a mole, and over time it begins to change and form into something different. That's why they tell you to keep an eye on your moles. But how dangerous is melanoma? Usually when found early, it is curable. There's a very high % rate of survival, but the longer you let it grow and don't notice it or go to the hospital and get it taken care of, the harder it is to cure melanoma. There are 4 types of melanoma, the first one being superficial spreading melanoma, then we have lentigo maligna, then we have acral lentiginous melanoma, and then the last one's nodular melanoma. Most of them are very similar. The superficial is the most common. Lentigo is usually found in older people. The acral lentiginous melanoma is usually found in people of color, and it's a lot harder to spot because it's found in areas that are not so well seen. And then the nodular melanoma is the most aggressive melanoma type out there, and it usually goes very, very deep into the skin, which makes it very aggressive. Next, we'll look at the epidemiology of melanoma. Epidemiology is an area of medical science that looks at how much or how little a disease or disorder is present in that area, and they'll compare it to other areas to determine why there is a lack or abundancy of that disease in that area. So for melanoma, white people have a 2.6% chance of developing melanoma, Hispanic people have a 0.6 chance of developing melanoma, and then Black or African descent have a 0.1% chance for developing melanoma. The most common areas where melanoma forms is going to be on the arms and legs. Looking further into the epidemiology of melanoma, we have mortality. Of the invasive melanoma, it's only 1% of all diagnosed skin cancers. However, it is 75% of all skin cancer deaths. The incidence of melanoma has been increasing, but the mortality rates have been declining over the years, as shown in a 2012 study in New Zealand and Australia. The current 5-year survival rate is about 93.3%, which is high compared to the 5-year survival rate in 1975, which was 81.9%. There's different survival rates depending on the stage of cancer that you're at. For stage 1 through 2, it's 99.4%, stage 3, it's 68%, stage 4 is 29.8%, and then of all the diagnoses made, only 4% of them are made in stage 4. The risk factors that lead to melanoma are usually sun exposure, indoor tanning, immunosuppression, moles, family history, and then obesity.
Staging for melanoma helps determine how serious each case is, depending on how much of it is present in the body. It's based on a T&M system that's focused on the tumor size and its thickness in the skin, and whether it's ulcerated, meaning whether it's an open wound or not. It describes whether the tumor has spread to lymph nodes and whether it's metastasized to distant organs of the body. The 2 main types of staging are clinical and pathological. Clinical being testing like biopsies or imaging, and pathological is pretty much surgical testing, which it helps determine whether the tumor can be removed or if it needs to be. So stage 0 is melanoma in situ, which is when it's just confined to the epidermis. It doesn't really spread any further than that. Stage 1, the tumor is no thicker than 2 millimeters, and it can be ulcerated, but there's no signs of spreading anywhere else. Stage 2, the tumor is 1 to 4 millimeters thick, and it can be more, but it's not ulcerated, or it can be ulcerated, but there's no signs of it spreading. And then stage 3 is when the tumor can be any thickness, but it can be ulcerated, but the cancer hasn't spread distally to different organs. And then stage 4 is when the tumor is It can be any thickness, it can be ulcerated, and it may have spread to peripheral lymph nodes, but it definitely spread to different organs of the body. So this slide just gives a good example of the different stages and where they are in the skin. So you can see stage 0, it doesn't go anywhere past the epidermis, the epidermal layer. It stays confined to the top. And then stage 1, you can see it's like a little thicker, but it still stays pretty much almost at the surface of the skin. It hasn't spread. Stage 2 is even thicker, but it still hasn't broken through that layer that allows it to reach into the dermis. And then stage 3, you can see that it has broken through that last layer and it has spread into the dermis, but it still hasn't reached the tissues yet, allowing it to spread distally. But this does allow it to, this is where it allows it to spread to like different parts like of the surrounding skin and the lymph nodes. And then stage 4, you can see that it has completely grown through the dermal layer and it's within the tissues, allowing it to spread throughout the body. And then the second picture on the right is just a photo showing early stages compared to advanced stages where they're both irregularly shaped. But you can see one looks more extreme and discolored than the other one. Yeah. So.
If we look at cell cycle regulation, P16 and P53 are tumor suppressor genes. P16 helps control cell division and P53 can induce cell cycle arrest or apoptosis. Surprisingly, P53 is not highly mutated compared to other cancers. It shows up 15 to 20% of the time according to the article titled Cellular Molecular Biology of Human Melanoma. P14 is also a gene that helps the cells from dividing out of control by blocking a protein called MDM-2, which destroys P53. However, if P16 and P14 are damaged, like it is in melanoma, MDM-2 can build up and shut down P53, even though it itself is not mutated. We've learned that FEGF stimulates blood vessel growth, and in the case of melanoma, this molecule is heavily stimulated, leading to angiogenesis. With more access to blood vessels, the cancer cells are provided with more oxygen and nutrients to further grow. Mutations also allow melanoma to avoid apoptosis. The cells will often produce high levels of BCL-2, which is a protein that prevents cells from undergoing apoptosis. This makes the cancer cells resistant to any neighboring signals and harder to get rid of. Melanoma cells will use glycolysis to increase their metabolic output even when oxygen is low. And even when oxygen is available, melanoma cells will still prefer glycolysis to support biosynthesis, which is considered the Warburg effect. There is an increase of GLUT1 that pulls sugar from the bloodstream, producing more energy in such a way that normal cells cannot. Cells also metastasize, aka they're able to move throughout the body. Normally, melanocytes are kept attached to the basement membrane. However, in melanoma, cells reduce E-cadherin expression, which causes melanocytes to stop listening to keratinocytes. Melanocytes become less attached and are then able to move. Melanocytes will then switch to N-cadherin, which normally is used by fibroblasts and blood vessel cells. The increase in N-cadherin also allows melanoma cells to stick to fibroblasts and blood vessels, allowing them to move out of the dermis and into the bloodstream. Growth pathways like FGFR-1 will also help the survival and spread of melanoma cells. Matrix melanoproteinases, or MMPs, will also help break down tissue barriers, which again allow melanoma to spread, changing the overall environment structurally. There are many ways that melanoma is being treated in a cellular manner. First is tumor-infiltrating lymphocyte therapy, or TIL, where the patient uses their own immune cells that are inside the tumor. This way, the T cells already recognize the melanoma, so the body can use and boost its own natural defenses. CAR T cell therapy is where CAR T cells are genetically engineered from the patient's blood. The T cells recognize a specific target on the cancer cells, almost like a GPS system, and go directly to the cancer cells. Dendritic cell vaccines are made by the patient's own cells and essentially act as guides of the immune system. They guide the T cells to recognize what melanoma looks like so the immune system can attack them. Lastly is adoptive cell transfer. TIL therapy is included in this category. These procedures have immune cells from the patient that are taken, modified, then given back to improve the outcome of the patient.
By looking at the molecular aspects of melanoma, we can understand how the cancer grows. First thing we'll look at is the MAPK pathway, which normally controls how melanocytes grow and survive. BRAF is a gene within this pathway that helps to regulate normal melanocyte behavior. But in about half of melanomas, a BRAF mutation causes the pathway to stay permanently turned on since it prevents melanocyte differentiation. Second thing we'll look at is PI3K AKT survival signaling, as melanoma does commonly activate this pathway. This happens when the tumor suppressor P10 is lost. Without it, the pathway cannot be kept in check, causing melanoma cells to resist cell death and increases their resistance to therapy. A key change that allows for melanoma is the disruption of the cell cycle. Through the loss of the CDKN2A gene, a tumor suppressor gene essential in making proteins that regulate the cell cycle, the breaks at the G1 to S checkpoint disappear, allowing for cells to continuously replicate even when they shouldn't be. The loss of TP53, another tumor suppressor gene that makes a protein that monitors DNA damage, prevents apoptosis from acting on damaged cells. So, through these disruptions, melanoma cells can keep dividing even with unstable DNA. Fourth molecular aspect we'll look at is telomerase reactivation. Typically, many melanomas have mutations in the TERT promoter, which reactivates telomerase. With a mutation in place, the telomerase keeps lengthening telomeres, allowing melanoma cells to divide indefinitely. Essentially, this causes them to become immortal. By understanding the molecular aspects of melanoma, there are a few treatments that can be used to target these molecular pathways to treat melanoma. First here is the targeted therapy, which would focus on blocking the MAPK pathway. By using BRAF inhibitors and other inhibitors, the overactive growth signal caused by the BRAF mutation is shut down. From this, tumors can be reduced. The second treatment is immunotherapy. Through the use of drugs that would allow for cancer cell recognition again, by stopping melanoma from shutting off important checkpoints, the immune system would be able to recognize and attack the melanoma. Both targeted therapy and immunotherapy would give a rapid response to reducing melanoma, but over time would lose effectiveness as the cancer would start to develop resistance. These approaches are typically long-lasting though.